And so while we've had some success, I do think that the future has got to be empirical. We are looking for this program to show itself in the next 6 months or so before the end of the year. We expect to be able to see what might happen and how we might go forward into phase two with which dose. We firmly believe that this combination should give us a better response than what has happened in the historical efforts with both chemotherapies and immunotherapy.
Welcome back and thank you for joining us for another Virtual Investor What This Means segment. Today we are featuring Sonnet BioTherapeutics, and I am very pleased to be joined by Dr. Richard Kenney. He is Chief Medical Officer of Sonnet. Welcome back to our platform, Rick.
Thank you, Janine. It's great to be here.
Before we get started, I just want to remind our audience that Sonnet BioTherapeutics is publicly listed on NASDAQ and trades under the ticker SONN. And during today's discussion, Rick will be making forward-looking statements, and I encourage everyone to view the company's latest SEC filings on their website at sonnetbio.com for the latest information. All right, Rick, we're going to dive right in, and just to set the stage for our audience. So Sonnet is currently enrolling a phase 1B/2A dose escalation proof of concept study of the combination of SON-1010 with atezolizumab or Tecentriq, which is provided by Roche Genentech in patients with platinum-resistant ovarian cancer. So my first question is, can you provide a background on SON-1010, the proposed mechanism of action, and why the company is exploring the combination of SON-1010 with Tecentriq?
Sure, Janine. We actually designed this molecule several years ago to target the tumor microenvironment, which should help with any immunotherapy. The way we do this is by binding to albumin, which is a natural molecule. It's the most prominent molecule in all of our bloodstreams that can target the tumor by going through the tumor vessel wall based on receptors that are on the wall. So we have the top end of a monoclonal antibody that we call the fully human albumin binding domain, the FHAB, to which we link an interleukin 12 molecule, the unmodified IL-12. I call IL-12 the conductor of the cytokine orchestra because it actually works on both the innate and the adaptive immune system. But if you link the two of those together and it binds to albumin, it rides as a hitchhiker into the tumor microenvironment. And there it can do its job to stimulate the tumor in all the cells that are resting there but are not exactly active. Tumors are interesting because once you stimulate them, you get interferon gamma as the primary output from this interleukin 12 stimulation. That actually upregulates the PD-L1 molecule. People have probably heard of that because that's what checkpoint inhibitors are targeting. We work, therefore, with Roche's atezolizumab, the atezo, because that blocks the PD-L1 molecule, essentially stepping on the brakes. And our molecule, the IL-12, steps on the gas to activate the immune system to work synergistically with atezo.
Appreciate that background, Rick. So, the company recently announced an update from your study, noting that a second patient had a confirmed partial response in E6 cohort of this study. This means that 2 of the 3 patients treated with the E6 dose have responded to treatment. Can you put these data into context for investors?
Sure. Anytime you're doing dose escalation, you're using small cohorts. So even though we technically have a 66% response rate, that's not terribly predictive of the future. In this case, we have an opportunity, though, to expand on that experience and have already enrolled the 15 people that we wanted to show the potential for the effect and are waiting for their responses. So the 2 out of 3 may not, resolve into 2 out of 3 out of the 18, but we expect several more partial responses out of this batch of patients that have ovarian cancer in a fashion that has not been seen very frequently. Most of the time with ovarian cancer that has recurred after platinum therapy, you get a recurrence in about 70% of the people. And that recurrence is treated successfully only about 20% of the time. You only get about 20% overall response rate. There's one study that may show a little bit more promise when the immunotherapy is combined with a pretty heavy dose of chemotherapy. But other than that, for 20 years, people have been trying all sorts of different approaches to do this without much success. We think that there's a pretty good chance in this case, given the history of what we have, to to break that historical precedence. We also know that as a monotherapy, our molecule SON-1010, which has this IL-12 link to the FHA-B, binds to albumin, goes to the tumors, has worked well in the other solid tissue space. And at this same dose, we've had 5 out of the 6 people respond with clinical benefit, and one of those had a partial response as well. So that's got an 83% clinical benefit rate in the monotherapy.
Wow, Rick, extremely encouraging. Congratulations on those data. My next question is, following an independent analysis of these data, Safety Review Committee recommended adding a new E7 dose of 1,500 nanograms per kilogram, a 25% increase from the 1,200 nanograms per kilogram dose in the E6 cohort. What is the company looking for with this new higher dose?
Sure, every time you do a phase 1 dose escalation, you need to try to consider both the potential for safety and the potential for issues. We've had incredible safety responses to date. We've had no dose limiting toxicities. We also look for the interferon gamma response, and at these doses are seeing a level of interferon gamma that we know is safe based on lots of history of other studies. We think that we could push this a little bit harder to get more interferon gamma and maximize the potential for success in this context with a little bit higher dose. 25% more of our molecule should give a concomitant increase in the interferon gamma, and that way we might be able to select the best dose for moving forward into phase 2, which is the next step after we see the results from this. Our safety profile has been so clean that we're eager to see what an even higher dose of SON-1010 can do.
Well, very encouraging, Rick. As you know, this is a What This Means segment. So, what do these data mean for your SON 1010 development program and overall strategy?
Yeah, I think you said the right word. It's encouraging. I try very hard not to have unrealistic expectations. And so while we've had some success, I do think that the future has got to be empirical. We are looking for this program to show itself in the next 6 months or so before the end of the year. We expect to be able to see what might happen and how we might go forward into phase 2 with which dose. We firmly believe that this combination should give us a better response than what has happened in the historical efforts with both chemotherapies and immunotherapy, and just are looking forward at the edge of our seat to see what will happen before the end of the year.
Well, we'll be on the edge of the seat with you. Definitely exciting times ahead for Sonnet Biotherapeutics and for sure the SON 1010 program. Rick, thanks for providing that background and all of that useful information for our audience. And with that, this concludes our virtual investor What This Means segment featuring Sonnet Biotherapeutics. And I'd like to thank Dr. Rick Kenney for joining us today. And I encourage you to visit sonnetbio.com for more information on the company and to follow their social channels to stay current on the latest information. Rick, thanks again for joining. Can't wait to have you back. And I wish everyone a great rest of your day.